T cells, also known as T lymphocytes, are crucial components of the adaptive immune system, playing a central role in recognizing and eliminating pathogens and abnormal cells. They originate in the bone marrow but mature in the thymus, hence T cell. Here's a breakdown of their structure, composition, types, functions.
A. Cell structure and composition. T cell receptor, TCR. This is the defining feature of T cells. Unlike antibodies that can recognize free-floating antigens, the TCR recognizes antigen fragments presented on major histocompatibility complex, MHC, molecules on the surface of other cells. Most TCRs are composed of two polypeptide chains, an alpha-alpha chain and a beta-beta chain. These two chains form a single antigen binding site. A small percentage of T cells have a different TCR structure made up of gamma-gamma and delta-delta chains. These gamma-delta T cells are found mostly in mucosal epithelial layers and can recognize a wider range of antigens, often in an MHC-independent manner. Each mature T cell has a unique TCR generated through a process called VDJ recombination, allowing the immune system to recognize a vast array of potential threats.
CD3 complex. Associated with the TCR is the CD3 complex. This complex of several polypeptides is essential for signal transduction when the TCR binds to an antigen MHC complex, triggering the T cell's activation. Co-receptors CD4 and CD8. T cells also express co-receptors that help them bind to MHC molecules and distinguish between different classes of MHC. CD4. Found on helper T cells, it binds to MHC class II molecules. CD8. Found on cytotoxic T cells, it binds to MHC class I molecules. Other surface markers. T cells also have various other surface receptors and signaling molecules, including chemokine receptors, which
T cells cannot directly recognize whole, soluble antigens. Instead, they require antigens to be presented to them by other cells, called antigen-presenting cells or APCs in the context of MHC molecules. Antigen processing. APCs like dendritic cells, macrophages, and B cells internalize antigens. Enzymes within the APC break down the antigen into small peptide fragments. MHC binding. These peptide fragments then bind to MHC molecules within the APC. Antigen presentation. The MHC peptide complex is transported to the surface of the APC. TCR recognition. The T cell's TCR then recognizes this specific MHC peptide complex. CD4 plus T cells or helper T cells recognize antigens presented on MHC class II molecules, which are typically found on professional APCs and present exogenous antigens from bacteria and parasites. CD8 plus T cells or cytotoxic T cells recognize antigens presented on MHC class I molecules, which are found on almost all nucleated cells and present endogenous antigens from viruses and cancerous cells. Co-stimulation. T cell activation often requires co-stimulatory signals like interaction between CD28 and its ligands to ensure a robust immune response.